Our next presenter is from SK Pharmteco. He is, I just want to mention, they are a 2025 CDMO Leadership Award recipient. That's a really big deal, and I'm going to tell you a little bit about that after this next presentation. Joining us is Nicholas Duda. He is the Executive Director of Business Development. Welcome, Nicholas.
Thank you, Barry. Thank you very much, and good morning to everyone as well. I'll get into some of the details here on SK Pharmteco. We are a small molecule API manufacturing contract manufacturing organization, but we also have alternate modalities in cell and gene therapy. We'll specifically talk about small molecule. Just to give a bit of a background about our company, we are working under SK Group. SK is the second largest conglomerate in South Korea, based in Seoul, South Korea, and they have an investment arm known as SK Inc. Under that investment arm, they have about 253 billion in assets and 23 billion in an investment breakdown. And you can see on the lower right portion of this slide, you have a bio and others section, and that's where we fall under as SK Pharmteco. SK Pharmteco is the sole contract manufacturing organization for cell and gene therapy and small molecule API manufacturing with sites across the globe. And that'll kind of get me into this side, a multi-modality capabilities in North America, Europe, and Asia. We have small molecule manufacturing in California and Texas. We have cell and gene capabilities in Pennsylvania, as well as south of Paris in France. And then continuing on, going across the globe into Dublin, where we have, again, small molecule API manufacturing. And then we also have two sites in South Korea, one in Sejong and Daejeon. Those sites in South Korea as well are for development and commercial API manufacturing. So just to kind of go briefly over the slides of how we work as a company, our primary goal as a contract manufacturer is to ensure that the medicines are available for the patients that we are treating. And we work to that goal, and we work with our customers closely, really kind of working to ensure that we're minimizing any type of friction necessary to be able to get to that common goal and ensure that the patients are being treated. We are very, very easy to work with. Our project managers will work directly with your project managers. Our technical expertise, whether they be chemists or engineers, will work directly with your chemists and engineers, coming up with engineering and chemical solutions to be able to keep the program moving forward. We are very, very flexible. We understand that timelines change. We also understand that scopes of work change. Maybe there might need to be some additional development required. We're able to work with you and come up with a new scope of work as necessary, as well as production timing to ensure that we're meeting your needs. We obviously pride ourselves on guaranteed delivery and strong communication. Again, looking at this to reiterate, when we are having technical conversations, we want to ensure that our technical teams are having strong communications with your technical teams. And that even gets into quality. We always operate on a high quality standard system. We are a CGMP manufacturing organization, and all of our API small molecule sites are CGMP manufacturing. And that CGMP manufacturing is coupled with our specialized services and our specialized technologies. Again, we pride ourselves on providing technical solutions using a course of technologies that I'll get into coming up here in another slide. So us operating under CGMP needs to maintain high quality standards. And what this slide really kind of indicates here is a long legacy of regulatory inspections across all of our sites globally. We have had 31 PAIs approved over the past decade, with 9 recent PAIs waived. And we've had 29 successful regulatory inspections since 2020. Currently, in our small molecule assets, we have 39 approved products in production. And this is ranging from scales of medium scale facilities to large scale facilities. You can see here, we have regulatory inspections from the FDA, European agencies, Health Canada. You can kind of see all of them there. We also have numerous customer audits throughout the year as well that we support. But the highlight that I really want to kind of recognize on this slide is our facilities in the United States have trained over 230 FDA personnel. Basically, these are FDA auditors that get CGMP training at our manufacturing facilities, and then post that training, then go out to the rest of the world performing their normal general audits or PAA audits. We pride ourselves on having an open door policy with the FDA, and we pride ourselves on being regulatory compliant and also teaching best practices with the FDA. So talking about some of our technical capabilities, I'd like to start off here with our chromatography capabilities. So obviously with every small molecule synthesis package that comes in, perhaps there could be some chromatography needs that align with it. We have simulated moving bed chromatography. This is a continuous chromatography method, as well as batch chromatography. In fact, in North America, we have a 5 by 1,000 millimeter unit. That's 5 columns, 1,000 millimeters or 1 meter in diameter for each column. And that is the largest CGMP unit in North America. That kind of flows into our continuous flow capabilities, whether you have fixed bed requirements, continuous stir tank requirements. Our facilities in North America and in Korea are there to be able to support any scale of continuous flow applications as necessary. And that continuous flow really came into our specialized handling of energetic chemistries that you see here, from diazomethane, azide, hydrazine chemistry, as well as ozonolysis chemistry. A lot of those energetic chemistry capabilities have a direct link to our flow capabilities that we have in the United States as well as in Korea. We also have high potent manufacturing that I'll get into a little bit more detail on later on. That's specifically in North America and in Ireland. Our Korea site has a very specific catalyst design group where we're able to specialize a catalyst specific for fixed bed hydrogenation capabilities. And we also have particle engineering capabilities at our Ireland site. Not only that, we also have milling capabilities globally. So we have milling capabilities for high potent API programs, but we also have milling capabilities for your general potency requirements, going from development scale all the way to commercial scale. And these are milling capabilities from hammer mills, Fitz mills, co-mills, getting all the way into Ventura mills that we work on specifically for our high potent. Again, all of these technologies are coupled with process development and scale-up optimization. When you bring a program to our facility, to our contract manufacturing organization, we pride ourselves on our process development, really scaling up those programs to be able to meet the initial clinical needs, getting prepared for the commercialization needs as necessary. And this is always coupled with analytical method development and validation. Each program that we work on, each chemistry program that we work on is always coupled with an analytical package that comes along with it. We pride ourselves in our strong analytical method development group that we have at each of our facilities to be able to support the programs. So let's get into it here a bit. And what we're going to start off with is basically SMB, simulated moving bed chromatography. Simulated moving bed chromatography is an excellent technique for the separation of diastereomers, separation of isomers. It primarily is a binary separation. Now I'll pause there on the technical detail, but would like to highlight on this slide that we have presented demystifying large-scale continuous chromatography for small molecules. Dr. Olivier Depremont gave a very good technical presentation on SMB technology, and I encourage anyone here to look at that QR code in the bottom right, take a look and be able to kind of find out a little bit more about the importance of SMB in your process development. With respect to scalability, our SMB units are in North America, and we can start as small as the 8 by 50 millimeter unit. Again, that's 8 columns. Each column is 50 millimeters in diameter, leading all the way up to the 5 by 1,000 millimeter unit, which we had discussed earlier. The scalability of SMB is extremely linear. What we're able to perform at the 8 by 50 millimeter unit can be scaled to whatever your demand is, whether it be initial kilo needs, whether it be some 100 of kilo needs, or whether it be multiple metric tons to hundreds of metric ton needs. We have all of that scalability within the United States. Sometimes SMB isn't always the solution, and we can kind of go and work with batch chromatography for complex separations as necessary. We have 50 millimeter batch chromatography units as well as 800 millimeter batch chromatography units within the United States, all dynamic axial compression columns. With respect to availability, if you bring a program to us, we can perform a quick screening, make a decision based off of whether SMB would be required or batch chromatography would be required, and within 3 to 6 months, we would be able to get onto the capacity that would be needed for that program. Again, proof of concept is established in the lab, and we're able to get onto a column within the 6 months after that proof of concept is done. Typically, our SMB is coupled with our semi-works units, and as you can see here, the Rancho Cordova site in North America has 5 specific semi-works units, Building 146 and the South Line, M&H, Building 41 and North Line. All are equipped with isolation capabilities, and these isolation capabilities typically go with batch processing post-SMB needs. You can look that we have hydrogenation capabilities across these lines, diazomethane capabilities across these lines, and you can also see the potency levels that we're able to work at. These range anywhere from 25 kilos to 400 kilo batch size needs. Our availability is within this year, and if you work with us, coming in the next couple of months, it's perfect timing for a start of a tech transfer and the ability to be able to work through the programs and scale them up. This then gets into additional pilot scales that we have in Rancho Cordova, where we're looking at anywhere from 5 kilos to 60 kilos need. And these, again, have availability starting in Q3 of this year and leading into 2026 as well. We also have large-scale facilities within our United States, are primarily here in Rancho Cordova. These are all equipped primarily with large-scale 2,000-gallon to 4,000-gallon reactor capacity, hydrogenation capabilities, as well as isolation capabilities. Again, these facilities do have open capacity starting in Q3 of 2025, and we'd be able to look at your larger scale needs to be able to support that. And these larger scale needs are also met in La Porte, Texas, where we have really large scale azide capabilities, as well as phosgene capabilities to be able to support your technical needs. This facility has openings starting in 2026. So let's take a trip across the pond over to Dublin, our Swords facility. Swords is located just outside Dublin, and I'll start with the pilot and semi-work scales there. To highlight here a bit, again, all of these facilities for the pilot and semi-works do have isolation and drying capabilities associated with them. Some of these facilities do have cryogenic capabilities, such as P7, and then as well as P4B. What I'd like to mention, this is an additional line that we're bringing on within the unit, and that's coming in Q3 of this year to help with our needs as necessary. I'll move on to some additional large-scale capacity that we have at the Ireland facility. You can see our P3 and P8 are booked, but what we are doing to accommodate that is bring on additional capacity with our P2 site, again, coming online in Q3 of 2025. This will be a 3-batch reactor unit, Hastelloy reactors, Hastelloy filter dryers, glass line reactors, Hastelloy filter dryers. And any of these facilities can be coupled with our bus loop hydrogenation capabilities. And that bus loop can take batch sizes up to 100 kilos to 125 kilos with availability within this year. Just to go into a little bit more detail about our high potent capabilities in Swords, our facilities are equipped to safely support high potent compounds. This is band 5 in quantities from grams to several kilograms. And this can be highlighted with our P4 facility and P4B and P4A facilities. Here you are looking at high potent API manufacturing with OELs of less than 0.01 micrograms per cubic meter. There are specific isolators associated with these facilities, and specifically P4B will be operational in Q3 of 2025. Some of the core technologies that we have associated with these facilities are TFF filtration. We also have lyophilization, and we also have some batch chromatography and flash chromatography associated with these high potent needs. Moving on to our South Korean site in Sejong, This is primarily where we have a good majority of large-scale manufacturing opportunities. We have pilot-scale opportunities as well. All are primarily linked with some of our fixed bed reactors, tubular reactors. A lot of them are linked with continuous applications. You can see that we have some of our availability in Q1 2026, and our module 2, which is the pilot scale, will be brought on from Q4 2026, but also some availability in the second half of this year in 2025. So as I had mentioned before, a lot of our programs are coupled with analytical requirements. We do also have a dedicated analytical service for your analytical needs with your programs. If you have no contracting of your API needs, but you need analytical services, we do concentrate on nitrosamine testing, extractables and leachables. So this not only goes into drug substance, but also drug product testing. We have stability studies that we are able to manage with our programs from drug substance to drug product. We also work on raw material and excipient testing. So if you have specific raw materials, we can be viewed as an overload contract analytical testing lab to be able to support those testing needs. We also work on reference standard management. So this deals with reference standard qualification, holding of reference standards, and also shipping of reference standards for your program as necessary. And just kind of dealing with some of the capacity expansions and our growth that we've established over the years. We have invested into our, in the United States, into our SMB chromatography and continuous flow capabilities. We have just brought on a high potent laboratory, which is now operational, an advanced high potent laboratory. And we've also increased 40% on our analytical capacity for stability testing and storage. Within Ireland as well, I had mentioned that bringing on our large scale small molecule facility that will be operational in Q1 2026, as well as our high potent kilo labs for ADC payloads. This will be operational, the P4B, P4A sections that I was talking about previously. This will be operational in Q3 of 2025. We also have a large scale small molecule peptide facility operational in 2027. This is major investment and major expansion in Korea. So I'd like to thank everyone for the presentation here. And Perry, if there are any questions or anything we can talk about, I'd be happy to ask that.
Thank you, Nicholas. Great job. Lots of expertise, lots of detail provided in your presentation. So thank you for all of that. Sure. Do you have one quality system? So you have a big global footprint. Do you have one quality system for every site?
Yeah, so we operate under one quality system across that. It's a global quality system. And basically that global quality system encourages best practices and the communication of those best practices across site. This integrated approach really kind of guarantees high quality APIs that are all within regulatory requirements specific for the clinical stage and the clinical needs that you have for your program. So yes, the quality systems that we have are extremely robust and practiced across all of our small molecule sites.
We have a question from an audience member, Ananda. So thank you for that question, Ananda. This has to do with SMB technique, excuse me, SMB techniques. Can you talk a little bit more about that technique with stereoisomer separation?
Sure, absolutely. So again, SMB is a binary separation, and typically what we use are chiral stationary phases. And if there are stereo centers that have specific chirality associated with them, we're able to go through a screening process that identifies that a very specific type of chiral stationary phase that works on molecules just like that. So we have a vast experience working with stereo center requirements, diastereomers, isomers. We also have a vast experience working on atrope isomers as well, and being able to work on separations at various scales for that. So we do have expertise, primarily doing a screening with specific chiral stationary phases to identify a separation.
So when it comes to scaling, and time is of the essence, nobody wants any downtime as the scaling process is ready to go. Can you collaborate across your various sites to keep that momentum going?
Yeah, absolutely. I mean, you can see across our sites that we have various scales from pilot to medium scale to commercial. Depending on your needs, if it's just a proof of concept where you're looking at initial kilo needs, we are able to start with that. But you can see any scale that you need, we're able to support, and the technical and engineering teams are there to be able to help with those scale-up needs as you're getting to larger quantities.
Awesome. Just a great presentation, Nicholas. Thank you for being with us today. Nicholas Duda from SK Pharmteco.